Again, we'll start. I have my colleague Sandra Sujin Lee on our at the top of our queue. And again, I want to invite people in the audience in the webinar to feel free to also type their questions in on the event page. So please, Sandra, jump in.
Oh, yes. Well, first of all, thank you so much for those presentations. They're very helpful. I had a question for Phil. Thank you so much for your presentation on the Million Veterans program. You mentioned that the identifiers that you're using are generated through self-report, through the survey, but also through that they're derived from the EHR and with the use of NLP. And I'm assuming that's information that's indirect, that that the latter is indirect information that you're trying to determine racial and ethnic identification.
That's correct. So usually that is information that is asked of the participant or asked of the patient, and then is inputted by the provider. But you're right. There over time, there's probably some assumptions made by providers, and maybe they put it in the information instead of the actual participant. So again, I mean, that's part of the beauty as well as the blemishes of working with a electronic health record, as well as in a large population like this, trying to harmonize this.
So I had two questions. One, just how do you do that work of deciding how do you harmonize when you have different information? But then the second question had to do with your hair algorithm. And it's really just clarification. What I heard is that whatever you have decided is the identifier from that first step, you then combine it with the genetic ancestry information to then bin them into the, I think it was 4 continental categories. Is that correct?
Yeah, so first of all, we don't try necessarily to quote harmonize the self-report information. We just catalog it and flag the ones that are not harmonized. And we use all of that data in conjunction with the genetic data. And so again, it's a machine learning algorithm that was trained on individuals that had both very congruous in terms of high probability of the genetic information matching their self-report information, and that as well as individuals that were incongruous and such. So I invite you to look through that manuscript and find out what the algorithm is. And most importantly, again, what we strive to do is be as transparent as possible and make the methods available, show the results, and all of this data, self-report from the EHR, self-report from the survey data, as well as the genetic inferred, as well as the hair, it's all available to our investigators and with the proper descriptions of how those data were derived. And again, where some missing data would be and such. And so we don't necessarily mandate the use of one or the other up until this point. And again, this is a dynamic process. This has been going on now since 2016 that we've made data available. So we have our ears open to understand what the community is suggesting in terms of going forward. And we again will try and incorporate that as much as possible in as best a transparent way as we can.
Yeah, so my question was really why you had to have one, why you needed to combine the information. Could you just keep them separate?
It was a choice to try, and again at that point in time, first of all, again going backwards in time, that this was an earlier stage where we didn't have as much genotype data, and so for individuals interested in power and such, we try to give them an option to have a specific number of race ethnicity bins to enhance their discovery projects. And so again, yes, we do have them separate, but we also, there was an attempt centrally to try and utilize as much of the available information as we had available to us.
Okay, all right, I might have some follow-up, but I'll give the floor.
Sure, absolutely.
Aravinda, you have a question?
No, I have a brief question of Phil as well, and perhaps the others could comment if there's time. I think partly many of the problems are at the nexus of not only identifying individuals in groups, but also because we are studying phenotypes. in them. I think one of the unfortunate things in genetics is we don't study families as well anymore, really, largely we don't. So I'm just wondering that in the MVP, is there any way in which you can get data on siblings? Because many veterans have unique exposures that the rest of us don't have. You know, Agent Orange is just one example. But my understanding is those kinds of data are not available on the veterans, at least not at the level of individuals. So if this is a population with very unique exposures, how do you think that you can disentangle them? Particularly if you don't have data on siblings, at least.
Okay, let me make 3 different points. Number one is as part of the baseline survey, we do ask about their family structure, and we have some of that information, as well as some information about family history of different health conditions as well. But certainly we don't have access to their electronic health records. Secondly, we do have families within MVP, or trios as well as some twins. And some of that study is possible. But I think the best answer to your question is that the fact that we have the ability to recontact these individuals. So if there are specific questions about exposures or bringing in family structure, we do have the ability to go back to them and have some contact with the participant first, and then secondarily, potentially with family members to do future studies. But it's not part of our current workflow.
Aravinda, did you want to?
No. Thank you.
Sure. John, please go ahead.
Great. So I thought these talks were great in bringing up some of the challenges of labeling and the strategies that come up and the practical decisions that are making, whether it's with the hair algorithm or some of the things that need to be context specific for working in Mexico. Alice, the graph-based approach that you highlighted at the end seemed very novel. Could you share more about that and introduce us to it?
Sure. Well, I can actually show you the beta version. We have it here. Hopefully that's, yeah, can you see that? Okay.
Yes.
So it is modular. This is the data set of the National Merit Donor Program. Sorry, not the full 13 million, but 100,000 roughly who filled out a survey, and they answered questions about their race using the OMB categories, but also about ancestry of themselves, of their parents and grandparents. And so what we have here is we have all of the categorical data that people gave us, but then also all the free text. So just everything that people put in there. And then we have a sort of very bare bones NLP processor that's reading in everything, and it's splitting everything up into single string text. And you can move it around. So the gravity, you can see it's not actually a 2-dimensional graph, but it's showing in 2 dimensions. This is the white OMB category. So there's 91,000 roughly people who selected that. And unfortunately, you can't see the layers right now because we're sort of, as I mentioned, still in beta version. But the idea is that you can essentially encode all of your aggregate level data as magnitudes of nodes and edges. So if I could show you this slide briefly, it might be a little more clear than this.
Alice, maybe introduce us to what an edge is. The nodes now.
Oh, I'm so sorry.
Text.
Yep. Okay. So thank you for that. So if you're thinking about, I sort of think about it like an accordion. So if you have an accordion and you expand it and you have slices of the accordion, each slice you could think of as a type of information. So let's say you have a layer or a slice of the accordion called race. Each text string of a race category, let's say, is a node. So this here is a node, and the size of the node is the magnitude of how many times it's been checked, let's say. So let's call these boxes. This is your categorical data. But let's say somebody checked other and then filled in the blank. the program will create new nodes to represent the information that people gave us. So rather than actually try to collapse all of the complexity of the data, we just collapse the data structure. So we don't have a spreadsheet with like African-American, 100,000 times. There's exactly one node in the entire data structure called American. And that one connects to African in one direction. It connects to Native in another direction, and so on. And then you have counts that are categorized in Python dictionaries that tell us how many times that node or that edge has been passed through. And that's essentially your sample size. So if you can manage to keep track of which data sets have been put through the server and not duplicate, then in theory, you can start to basically build a graph-based data structure that gives you a prior probability of relationships between categories. And so you can add any kind of data you want for any layer. And then you can actually look at the relationships in advance and then use sort of Bayesian statistics to look at your differences between your priors and your posteriors. It's because allele frequencies, as we're calculating them now, have the denominator is the number of people in the category. So the problem is like, well, if you have a million categories because people are putting in all different information, how do you calculate allele frequencies? Well, I'd argue you don't have to do that. What you can do is you can use the number of observations of that allele as a denominator in your frequency. And then you can look at the distribution across the categories. And you sort of expect that the observations are going to map with the distribution of the individuals across categories. But if you have enrichment or depletion of the observations relative to the individuals who selected those categories, then you can start to compare frequencies in sort of a different way.
Fascinating. Okay. Yeah. So in the map you showed us earlier, the edges were like co-occurrence of a word in a record.
Yeah. So an edge can be a couple different.
But there's also across these layers, I guess. So there's two notions of edges. Yeah.
Exactly. So I call them paths if it's representing an individual. And the beauty of this is that you can keep the edges and the edge weights within a layer, because that tells you about, how many times somebody is Black, African-American, and American Indian, Alaska Native. So it'll look like one long path connecting all of those individual nodes, and that's a person. But then you'll have aggregate counts that you keep. And then you can either elect to keep the paths that connect all of the different variables between them, or not. And the way that we have it set up right now, we have a portal that we're going to launch at some point where people can actually upload their free text data once they check other and fill in the blank. And then we'll tell you what layer it maps to. We're not going to change it. We're not going to impute anything. We're just going to tell you how many people in your sample gave you information about their racial or social identity or their ancestry, that kind of a thing. And when it maps through, then it'll keep the aggregate counts, but the researcher should in theory be able to decide whether or not they want us to keep the individual paths, or whether we just keep the aggregates, or whether we just keep the nodes. And also we want people to be able to run it on their own servers, so it doesn't ever go over the internet. So we want to respect Indigenous data sovereignty, for example, and enable people from a mechanistic perspective to use and benefit from the software without actually having to give us their data, because we hope enough people will want it to be a public resource that they will.
Great, thank you.
Great. So we're still open for questions from the committee, and since I don't see any hands up right yet, I'm actually going to take advantage of the queue to jump in with a question for each of our speakers. So first of all, for Phil, I wanted to know simply, I was very struck to see that you are basically using the OMB categories when you ask people to describe themselves. So you're using the same categories as the census uses. And I wanted to simply ask, are you mandated to do that? Is the VA required to do that? Or would you have flexibility in tweaking those categories?
I don't think we're mandated. I think because those baseline surveys were developed before I joined the Million Veterans Program. And if I remember historically, that was based on traditional epidemiological studies, which again, use the OMB categories. So we've talked about enriching those categories, but you can imagine the challenges of trying to decide what questions to go back to our participants on a scale that we're at right now. So we are open to it, but we want to do it in a metered and thoughtful way.
Okay, all right, thank you. So my two other questions, I'll ask maybe Michelle first, but give you a second to think about it and then put a question to Alice and come back to you. Michelle, I just wanted to ask you because, I had you, ripping through the presentation on time. If in a rush at the end, if there's anything that you'd want to come back to that you didn't quite have enough time to flesh out for us, but that you would, like to maybe underscore as a major takeaway from it that you may not have had the time to devote to that you'd like. And then with respect to Alice, maybe I'll ask you to answer this question first, Alice, and that is just looking at the work that you showed us from the National Marrow Registry. So that's a US-based database. So it's got labels in that presumably a US-based sample has put in. Do you think that this method that you're showing would be of help actually in thinking about harmonizing across national populations? That is, with this way to empirically detect relationships between different kinds of text streams, different terms, would this also be something useful if, for example, we wanted to bring together data from the US, Brazil, and France, just to take, a series of countries with very, very different approaches to categorizing and to self-identifying people? So maybe, Alice, I'll start with you and then come back to Michelle, if that's all right, and then pick up some questions in the queue.
Sure. Yeah, thanks for the question. I think in theory, it's definitely flexible enough. In practice, it'll be challenging, I think, for people to break out of the categories that their societies have sort of molded them into adopting, right? In Brazil, it's like very shocking for us to see yellow as one of the racial categories, but I'm sure ours seem just as arbitrary and shocking into other cultures. But in practice, the structure itself, the data structure, is designed so that people can use their own words, their own language to describe themselves, and it will preserve what they've told us. That's, I think, the key, is that so much of the methods we use currently are designed to collapse and reduce the dimensionality of the data. And I think we can reduce the dimensionality of the data structure, or reduce the complexity of the data structure and the size, while still allowing for the flexibility of whatever people want to say.
Okay, thank you so much. Michelle.
Sure. So I think one thing I would just say is, I think some of the things that's come up is related to which descriptors to use and why. And I think there seems to be a promising direction in using both genetic metrics, right? So say some of the proxies that come out of these methods, like admixture or PCA, or newer methods that would truly have a more continuous sort of output coming out of it, and using simultaneously, say, race in the same model. Because what race will give you is how much of the health disparity is because of the lived experience of race and discrimination and the resulting health impacts of that. Whereas the genetic metric that you have is, while still capturing any residuals that you're not capturing of the lived experience, is capturing some of the things that I was trying to say off, that there are patterns of genetic variation that do differ because of these different ancestries, different demographic histories, right? So I think we can try to just model the two together with respect to trait variation, and doing that, we can try to separate how much of the disparity or variation we're seeing is coming from racism versus maybe coming from specific patterns that come out of demographic histories. And again, even with the part where it is coming from the genetic patterns there, to do that on different scales, right? So it could be patterns that are more recent demographic history, or older demographic history, or archaic, right? So, and really showing because any of those can be important depending on the trait that we're interested in, and to not just sort of really only highlight the continental ancestry slides, which is just one-time slides. So I think it's super promising with these mixed models and using both kinds of predictive strategies to disentangle the two effects. Because what I see a little bit is from more humanities and social science circles, it's just disparities are definitely due to racism. And within genetics, there's a lot more interest in trying to find genetic markers of that. And maybe there's a place where we can try to separate the two by working together and making models that can take both kinds of variables.
Thank you so much. And maybe I should have mentioned to the audience that you're rather uniquely positioned to sort of speak to the work that both folks in the social sciences and humanities and in the biological sciences are doing by virtue of your own graduate training, also on some of the humanities side. So thank you. Aravinda, I see a question from you.
Yeah, so just as a member of the committee, Alice, I think some of the points some of the aspects that you spoke about, which may be a way out, I think require a much deeper discussion. So hopefully we can connect even as a committee after this. So this may not be the last. But talking of this dual exposure and that you mentioned about Mashal, I wanted to ask you a question of having worked both in the US as well as now in Mexico. So race and ethnicity are the two strong terms that we both use and react to in genetics. So have those terms been helpful when you've tried to address the same kind of biological problems in Mexico? How helpful have the terms been? How helpful have the constructs been in sampling people, describing people, using them in genetic studies, comparing your results to others, which is ultimately what we want to do. There may be good reasons to use them, for example, in studying the effects of racism on health and health disparities, but were they necessary?
It's a great question. So I think one thing that's just become even clearer than it was before by working and coming to Mexico is just how much of a social construct race is, right? Because just like being Black means something completely different in Mexico versus in the US in terms of definition, in terms of its correlation with genetic ancestries. So I think in the context of Mexico, I had to sort of think carefully and deeply about the particular histories here, right? Which is very different from the histories in the US. And so here there's a specific colonial context where there's more of a divide between indigenous identity versus more European identity, right, versus white Black. And so I think it was helpful to learn about those different indigenous identities in different places and to make that part of the project, along with thinking about the ancestries that have come from different parts of the world. But I think where I struggled really was with the realization that when you do the ancestries from the continental level, they're very much mimicking the ideas of older racial categories, which Alice also gave much of a prelude to, about how, like the fact that there's like sort of 5 groups or so on, and, older ideas about that, how they're existing a lot in the hierarchy, like that, our continental ancestry ideas can be mapped onto that. And that's what keeps troubling me with the project. So that's why it just became important to me to use other resolutions, other timescales as well, so that it's certainly not just about that one time slice. But I think where the struggle remains is like, I do see how that can be relevant genetically and therefore potentially for biomedicine, because I do show and see that these different proxies of continental ancestries that we get from something like admixture and global populations do reflect like different population size histories, different patterns of ROH, and so on in a significant way. And I can very much see as a geneticist how that might be relevant for diseases that have more recessive component and so on and so forth, right? So yeah, I don't feel like I have the answer. So I think in this project, I just tried to do as much as possible to problematize and show the complication. But I think it's very clear to me that there's some relevance of thinking about demographic histories, right? More and more, I prefer to use that, or ancestries versus, sort of.
I did not mean that as a criticism. I just meant, really, what the challenge of studying type 2 diabetes, of comparing results, say, of example of type 2 diabetes in Mexico versus in the US, or say even across the border in Texas. So anyways, we may also come back to sort of your thinking on this and thinking about
Okay, thanks.
Thanks. I think we have time for just one more question. John, go ahead.
Great. I guess this would be a hard one to answer quickly, but if you can try. From where each of you are sitting in terms of what would move the needle and like shift your groups or your colleagues and collaborators to adopt new practices in this area? What do you think it would take? Because that's one, I think, the real practical public policy making aspects that we need to think about as a committee.
And each of you has 30 seconds just to make it
Well, one
even
of you can
easier
jump
for
in
you,
and
right?
make-
No pressure.
Well, I'll just say, I mean, obviously incredibly challenging, and I think that each of us as a cohort needs to be open and be listening to what the community is saying in terms of how they are deriving these different methodologies and conclusions. And I think it was Nancy who said in the earlier session, it's a dynamic process. And again, we have to be ready to be able to change in a way that is practical, but at least as well as informative and sensitive to our participants, which is of highest priority to us.
Thank you.
I'm cheating, John. I just shameless plug for the recommendations and the thoughts that came out of the ClinGen Ancestry and Diversity Working Group, since I didn't have time during the talk to go over them. I think the basic gist is that there may be some uses for population descriptors necessary for the question you're asking or to track diversity. And obviously we have requirements by the NIH, but that it's not really effective when you're conflating different types of information, when you're reporting allele frequencies or comparing these point estimates across categories that have different, sizes, and that categorical frameworks essentially fail to represent global diversity. So that we should really be thinking about how to do this in a way that's more globally representative.
Thanks.
Okay, that's really helpful. Michelle, you got the final word.
So I think in my case, like I'm having students in the lab work on this project using existing groups from Thousand Genomes AGDP to like really look at classifications and to really question them. And I think that's really helpful because like they see, for example, if you look at singletons per group, like they don't order groups by continent, they order them across beyond continents, right? Or if we really look at admixture analyses in a careful way, like they can clearly see like how much it matters what we put in with respect to what we get out, to understand that the K is not some innate property of the world. It's completely dependent on analysis decision. So I think in that light, like what I think it can come out is to be very, very clear with yourself and your team why and when you're using a metric. So if you're using race, like what you hope to get out of it. If you're using genetic ancestry, what you hope to get out of it. And in that sense, being able to combine these as necessary, being super clear about when sometimes, like race is not the relevant metric. Sometimes like if two populations that are far apart in Mexico are more different in some trait that you're interested in than thinking about European versus native, right? So being very just careful about that and not always thinking that those continental categories actually divide us up genetically. So showing in a data-driven way, which I think more and more studies are showing that they don't divide us up that way. There are different metrics bring out different classifications. It may be relevant for different sites of biomedicine.
Thank you so much. And again, I'd really like to thank so much the speakers who took their time to share their expertise with us now. We'll now be moving into a break and we will resume at 4:20 p.m. So thank you all.